There guys, this is Mr Phillips doing another video on A Level Biology. So this is year 1 video, and this is video 1.4 I believe, which is Structural Polysaccharides. So what we did in the previous video is look at starch and glycogen, which are energy storage polysaccharides, but these ones are structural today. So what we're going to be looking at is really the structure of cellulose, and what you can see here, this man putting in a sustainable house cellulose walls, which shows you how strong cellulose actually is. So hopefully by the end of this lesson you'll be able to do explain what the structure of cellulose is, particularly explain how it's adapted to doing its role, and also just relate it to any roles it has in particular. So just to remind you, because you need to know this then, that alpha and beta glucose, so if you remember, if you watched the first video I did, you will remember I would have told you about the difference between alpha glucose and the difference between beta glucose. So alpha glucose, it's all about what's happening at carbon 1 of both of the isomers of glucose. If you notice, just for the record, for a few minutes time, carbon 4 on both of the molecules is exactly the same. So carbon 4, key thing is the hydroxyl group, which if you remember is what helps bond things together, is on the bottom, okay? But on alpha glucose, okay, so this one here, okay, on carbon 1, the OH group, the hydroxyl group, is below. So in alpha, it is below, and in beta, the OH group is above. Hence the term ABA, alpha below, beta above. Okay, and it's all about those hydroxyl groups which form the condensation reaction bond, sorry, the glycosidic bond between sugars. So going back to previous lesson again, with alpha glucose, so today we're going to be looking at beta glucose, but with alpha glucose, which makes up starch and glycogen, it's a pretty obvious process by now. So you have here the two OH groups that are aligned next to each other naturally. So what happens is, if you follow the blue dotted line, 2 hydrogens and 1 oxygen comes out. It's a condensation reaction. A water molecule comes out, leaving 1 oxygen to link the two together, because it has a valence of 2, which means you can have 1 bond to carbon 4 there and 1 bond to carbon 1 there. So because that's carbon 1 and because that is carbon 4, we call this a 1,4 glycosidic bond. Okay, so 1,4 glycosidic bond. And when we break that bond, we add water into it in a hydrolysis reaction. However, it's not so straightforward when it comes to beta glucose, and the reason is, if we look again at what beta glucose is, so remember in the saying ABA, alpha below, beta above, on carbon 1 of beta glucose, the OH group is above. So if we've got 2 beta glucose molecules, the OH group is always going to be above. On carbon 4 of beta glucose, which is exactly the same as carbon 4 of alpha glucose, the OH group is below the carbon 4. So what that does is it gives us a bit of a problem, because no longer do we have a OH group here, because if we had OH here, we'd be able to join an OH group and an OH group together to form the 1,4 glycosidic bond again. But unfortunately, we don't have that. So there is no bond that can form between that hydrogen and that hydroxyl group and that one. It also is too far, or the enzymes don't have the correct shape to link up OH there with OH there. So that can't happen either. So somehow we've got to try and get these two OHs together in order to allow us to have a 1,4 glycosidic bond. So how do we do this? Well, how we do this is actually relatively simple. Now you've got to remember these are 3D shapes. So when I'm saying twisting them, we're not necessarily twisting them like that. We're sort of twisting them like that. Okay. So what we need to do is we need to take these molecules and we need to rotate them. So if you look at this one, we've got here at the bottom of the screen, sorry. Okay, so if we look at this beta glucose molecule, okay, first of all, this one is the correct way up. Okay, so it's facing upwards as you would expect. Okay, so that means this is carbon 1 and this is carbon 4, as we've said before. Okay, but because it's the right way up, bit alpha below, beta above, so the OH group above carbon 1 is above. Okay, this one, however, if you look at what the difference is between this beta glucose, the molecule is facing down. So exactly the same as before. This is still carbon 1. Okay, this is still carbon 4, but this time, as I just said a moment ago, it's been rotated like that. Now what that means is because this one is facing down, this one's facing up, it now allows us to have an OH group which is below carbon 4, but obviously below carbon 4, carbon 4 has now been switched around. So what was down here is now up there. And what that allows to happen now is that the carbon 1 hydroxyl group and the carbon 4 hydroxyl group have aligned, and we can now have a reaction where we take out the water molecule, okay, leaving us then just with an oxygen. So if we go on to what this then looks like, you get a chain of beta glucose molecules. So if you have a look at this beta glucose molecule, okay, this one is facing the right way. Okay, this one has been rotated 180 degrees down, and this one again has been rotated 180 degrees, so it is the right way up. So if we look at the carbon 1, okay, so remember we've got carbon 4 there with the OH group below as normal. Carbon 1 here, the OH group will be above because it's beta glucose, you'd add your OH here. Okay, then the next molecule has been rotated 180 degrees. So on this carbon 4, the OH group is now here. Okay, and that means that those two are able to make a hydrolysis reaction, sorry, a condensation reaction happen, and you get your oxygen that links the two up. Now, because then the next one is rotated 180 degrees again, this one was upside down, so the OH group is above carbon 1, but it's been rotated so it ends up below the structure, so it's hanging upside down if you imagine like that. This one, the OH group, is below carbon 4, okay, so therefore they are able to do a condensation reaction. So what this means then is this allows beta glucose to form long, strong chains, okay? And effectively, this allows a different characteristic compared to when alpha glucose forms chains. So if you remember with starch, you've got long spiraling structures. The alpha glucose, because of the way it bonded, this doesn't happen with beta glucose. So a lot of students struggle with how to write this in a way that is going to get them the marks in an exam. So I've used an exam mark scheme here to try and highlight what you should write. And the key thing is your description of the rotation of beta glucose. So if you look at this box, it just says by rotating the beta glucose 180 degrees to the last one or to the previous one. Both of the hydroxyl groups, and I would always write hydroxyl if you can be aligned. Okay, and then that just links you back to the other thing. Then you can remove water via a condensation reaction, and it creates again, and I probably should have wrote there, a 1,4 glycosidic bond, as we saw a moment ago. Now the difference is this time is because of the fact that one glucose molecule is the correct way up, and the next glucose molecule is the wrong way up, and the next one is the correct way up, and they've all been rotated 180 degrees to the previous. It means the shape becomes different, and the bonds don't form a sort of don't cause the spiral shape as they bond. So you get long straight chains of beta glucose forming what we call one cellulose chain. So this whole thing is one cellulose chain. It could be 1,000 chains long. And as a result, cellulose is one of the most abundant structural polysaccharides there is, because obviously cellulose makes up the cell wall. Now, hopefully you know the cell wall needs to be strong, and it needs to be strong because it needs to withstand water pressure, so cells can become turgid. It needs to be effectively the structural element of the cell, keeping the cell in its structure, but also in cells like guard cells, there's extra cell walls, extra cellulose layers, which will help maintain the shape and help the opening of the stomata. So basically what we have is like a multi-layered system, like rope effectively. So if we start off, this was our beta glucose chain, which was just like from the last one, so one up and one down and so on and so on. Okay, what that does is, so imagine that is one of those chains. Each one of these is one beta glucose chain here. They all combine together, and so all of these combine together to form what we call a microfibril. Okay, and then lots of microfibrils combine together to form what we call a macrofibril. And what happens is there's two ways that these molecules are held together. So the first way is between between beta glucose chains or cellulose chains, there is hydrogen bonding happening. So hydrogen bonding, we've done a different video, and we'll do on the water video as well. But hydrogen bonding is weak forces between the OH of one group and a hydrogen of another group. So you can see there is a hydrogen bond, there's a hydrogen bond, and because they might be very weak individually, but there might be a million hydrogen bonds in a long chain, it makes it a lot stronger. It means the layers of the cellulose can't slide over each other, as you might expect. Now, on top of that, within this structure of beta glucose and beta glucose chains, there is a natural glue called pectin, and pectin basically fills up all of the gaps and any of the gaps in these layers, and then when it obviously dries out, it glues everything together, giving cellulose that extra strength needed for its role. So that's it for this video. Some key information here for you to answer, see if you can answer these questions. Question number 4 we haven't addressed yet, but just briefly to let you know, there are two types of other structural polysaccharides you might need to know the name of, and that is in insects there is a molecule called chitin, okay, and in bacteria cells, the cell wall of a bacteria cell is made up of something called peptidoglycan, which you can have a look up at your own time. And the last bit, the extension question, links to GCSE. Why would we be able to digest alpha glucose as humans but not beta glucose? And if you think about it, it all links to enzymes and active sites. So I'll leave that one with you to look at. Thank you very much, and I'll see you again soon.